Tight Junctions by Regina Tanchez. Tight junctions are an area where the membranes of two adjacent cells are joined together to form a barrier. The cell membranes are connected by strands of transmembrane proteins. The most important functions are to help cells form a barrier that prevent molecules from getting through and to stop proteins in the cell membrane from moving around and holding the cells together. It also helps maintain the polarity of cells. The tight junction acts as a permeability barrier in which proteins are organized in continuous strands in the plasma membranes around the necks of each adjacent cell. The tightness of the barrier is affected by the precise composition of the junction, and it varies with the tissue type and can be regulated to allow the passage of certain molecules or ions depending on the local environment conditions. Often found at epithelial cells, which are cells that line the surface of the body and line body cavities. Not only do epithelial cells separate the body from the surrounding environments, they also separate surfaces within the body. This is why it is very important that the permeability of the molecule through the layers of the epithelial cells are tightly controlled. Epithelial cells, which is required to separate tissue spaces and regulate selective movement of solutes across the epithelium. Tight junction integral membrane proteins are comprised of claudins, occludin, tricellulin, junctional adhesion molecules, also known as JAMs, and Coxsackie and adenovirus receptors, also known as CAR. Claudins and occludins are the two main types of proteins present at tight junctions. Claudins are important in forming tight junctions and constitute the basic framework, while occludins play more of a role in keeping the tight junction stable and maintaining the barrier between cells that keeps unwanted molecules out. Tricellulin is a four-pass transmembrane protein that is concentrated at the vertical orientation tight junction strands of tricellular contacts of the epithelial cell sheets. JAMs are involved in the barrier function of tight junctions in both endothelial and epithelial cells. CAR contributes to the barrier function of tight junctions in epithelial cells, but not in endothelial cells. In the top image under epithelial cell, we can see in the image what looks to be like a blue net. That is the tight junction, and to the right of that is the tight junction integral membrane proteins and the structures that they have. Underneath that, the image shows an arrow where the arrow is pointing is the tight junction within a mouse. Tight junctions are primarily located in the epithelia, which is the body's internal and external surfaces such as skin, blood cells, and cavities. The most important functions are to help cells form a barrier that prevents molecules such as ions, water, and various macromolecules, even cancer cells, through paracellular spaces, from getting through and to stop proteins in the cell membrane from moving around and holding those cells together. It also depends on the location of where the tight junctions are located. So like let's say the skin, where it keeps us somewhat watertight and helps keep allergens out of the body. In the digestive system, it prevents the leakage of digestive enzymes into the bloodstream. And in the epithelium, structural support mechanisms that help keep it together. Epithelial barrier dysfunction and inflammation in inflammatory bowel disease is genetically encoded variation in the epithelial barrier function, and it may allow microbes to cross the barrier and trigger a T cell response. In the number 1, the cytokines produced by the activated T cells in the macrophages loosen tight junctions, allowing more antigens to cross. And in 2, we finally see the degradation of the basement membrane, which causes the epithelial cells to shed and a massive penetration of microbes into the gut wall occurs. In recent findings, they have found a new way to peer into the protection connections or junctions in animal cell cultures to directly measure their properties. Scientists in the past have lacked effective ways to directly measure the junctions between epithelial cells, and the previous studies were really crude and required damaging the cells in the process. In this new study, the researchers modified a method called non-contact acoustic frequency modulation atomic force microscopy, also known as FMAFM. With FMAFM, a tiny probe is brought close to the cell and it then gently vibrates, not causing any damage to the cell. The researchers here created a new theory so they could use this method to study previously unmeasurable properties and forces between the cells. In this research, researchers compared normal cells to cells lacking proteins called ZO1 and ZO2. These proteins are important for tight junctions. And using this new version of FMAFM, researchers could measure the increase in tension. Moreover, using these tension measurements together with information about the hexagonal shape of the cells, they could explore the forces between the epithelial cells and the junctions between them. My question is, what is the difference between desmosomes and tight junctions?